Hello again, and here we are with the nervous system. In this lecture, we're going to focus on microanatomy and the fundamental functions of neurons and a little bit with glial cells. We will deal more with the macro structure next time in class, but this time around, we're going to focus on the microstructure, the cellular structure, and the function at the level of the cell, which is where the action is anyway. For instance, some of these questions right here are very, very reasonable in terms of how to think about some of the ways that the neurons and the glial cells behave. For instance, what is myelin? Myelin is a type of lipid. All right. You know what gets on my nerves? Myelin. Okay. It's an insulating lipid that prevents unwanted ion exchange. And we'll show you how that works. It's a myelin sheath. What does it do for the neuron? It's an insulator. It helps prevent the decay of an ion mediated stimulus. And again, we have some figures to show you that. What the heck is a node of Ranvier? Node of Ranvier is the spaces in between the myelin sheaths, and that's where we get action potentials when we talk about saltatory conduction. And another one, this is more on the macro level. What is white matter and what is gray matter? Well, when we look at a histological preparation of, like, for instance, a spinal cord, or you could look at the brain or mostly central nervous. You don't really see much in the peripheral that way. The gray matter, I'll start with that first, is where we find lots and lots of neuron and glial cell bodies. So we see lots of nuclei, and it stains a little bit darker, which is oftentimes with the original stain, it was a little more gray because of the nucleus and because of cell bodies rather than myelin. So they called it gray matter. Then we have white matter, which is mostly myelinated axons. There are a few cells in there. You're always going to see a few nuclei here and there, but it's predominantly lipid. So what we see are axons sheathed in myelin, very few nuclei. So that's why we call it white matter, because it generally stains white. So here we have some examples of myelinated axons. The axon is here, and surrounding that axon are these little segments, look like sausages, of myelin. And myelin, again, is that lipid that serves as an insulator. Now, this figure shows what we call a multipolar neuron here. There's another one and another one here. These are downstream of this first one. And we have a lot going on in this picture that's not really illustrated by the text here. For instance, we call it multipolar because it has one pole, that's the axon here, that counts as one. And then we have several other processes here that are known as dendrites. Dendrites are the input surfaces among, you can put contacts with other neurons anywhere on this cell body that is not myelinated here. Okay, but in particular, we see lots of contacts with dendrites. Dendrites increase the opportunity for neurons to contact. And you can see this presynaptic terminal is touching this dendrite, and this one's over here, and this one's touching here, and so forth and so forth. So there's a lot going on here beyond just myelin. But if we get back to myelin, in the central nervous system, the cells that make these myelin sheaths are known as oligodendrocytes. And I'll show you some pictures of those here in a little bit. And in the peripheral nervous system, this is much more common in lower level anatomy and physiology courses. You have what are called Schwann cells. Some guy named Schwann first identified these cells. And we find these outside of the central nervous system. So we find these beyond the spinal cord. The central nervous system is the spinal cord, the brain, brain stem kind of thing. Everything else outside of that is peripheral. So we see Schwann cells in the periphery. So with that little introduction, let's actually break out how we're going to approach the material for the rest of the lecture. We're going to look at the basic structure of the nervous system. So we will do a little bit of macro here. We'll do a little bit of macro anatomy. talk about the different divisions and the different ways that we can classify the nervous system. You can classify it by location. You can classify it by direction. Is it moving towards the central nervous system or away? And you can classify by function. So we'll break some of these terms out for you. The types of glial cells, you can also say neuroglial, but they are glial cells. These are the non-neural cells that we find associated with neurons, and they are absolutely essential for proper neuronal function. We have different types of neurons, and mostly in this case, we're gonna show these by shape. You can also classify neurons by certain kinds of function, but and tied in with that, you can also classify them by the neurotransmitter that they secrete. Like for instance, you can talk about dopaminergic neurons. These are neurons that we find that secrete dopamine, one of the neurotransmitters. But we will mostly stick with shape here. We're not going to break out neurotransmitters all that much. We are going to get into the cellular structure, especially of a multipolar cell. The multipolar cell is kind of our poster child, if you will. It's kind of what we're going to use as our reference point. There's no point in getting into much of the other ones other than just understand that the neurons do have different shapes, and that sort of translates into function a little bit as well. We are going to get into resting membrane potential and how that's established, the ionic sources, that is sodium and potassium, being pumped across the plasma membrane, and then we allow a differential leak. In fact, the differential leak is the basis of all the signaling, okay? It really comes down to that. We start changing the permeability of the membrane by opening and closing channels, and we change the voltage, and we use that as a basis for signaling. But we're going to start with resting first, and that's the magic number there, minus 70 millivolts. That's because we pump the ions, and then we differentially allow them to move back across the membrane. We are going to get into action potentials, particularly the rising phase, known as depolarization, the falling phase, known as repolarization, and then this reset phase. That is, the channels are resetting and the cell is kind of getting back to resting membrane potential, but it overshoots. It gets a little bit more beyond minus 70. It moves into minus 80 or minus even beyond that, where it's hyperpolarized. And that's when the channels that set off the action potential, these depolarization and repolarization, it reset and allow the neuron to fire off again. Within that hyperpolarization phase, we have what are known as absolute and relative refractory periods. The word refractory means resistant to additional signal processes. If it's like, for instance, if you have a refractory cancer, that means that the cancer is resisting whatever chemotherapy or radiation therapy you're giving at it, okay? So we have an absolute, which means that there is no getting through that refractory period. And then there's a relative, which means it's maybe, but the signal has to be stronger. Yeah, I'll cover that a little bit later, but I want to get you started early. We will know about how to send a signal across what is known as a synapse. We have electrical synapses. We'll talk about that for just a moment. And we have chemical synapses, and the chemical synapses are much, much more common. These are the ones that use a diffusible chemical known as a neurotransmitter, all right? So we're going to talk about how we get that neurotransmitter out of the presynaptic terminal, how it gets across the synapse, and how it affects the downstream cells. We'll show you some pictures. Looking forward to it. Here's just one direction where this can go. And this is very, very common, especially for folks who go into nursing. Oftentimes, the procedures require some diminishment of sensation, and we call that diminishment anesthesia, the controlled loss of sensation. Anesthesia, the sensations that we perceive are neural mediated. So the neurons are a very opportunistic target for blocking sensation. There are ways of blocking the initial signal. Okay, there are ways that you can block up there are other, you can block the original signal, but neurons make the most druggable target most of the time when it comes to anesthesia. So we have local anesthesia. This is where you have loss of sensation in the area that you're applying that anesthetic, but you don't have loss of consciousness. All right. Lidocaine, Marcaine, the canes, if you will. Cocaine falls into this a little bit too, but it's kind of complicated. It blocks what are known as voltage-gated sodium channels that are involved in action potentials. We will show you that, and I'll try to remember to tell you about this is where the lidocaine works. Okay. And we have what is called general anesthesia. And what you're doing there is you have loss of sensation that is usually secondary to loss of consciousness. Okay, that general means that you're knocking everything out. Okay, mostly what we're thinking about here is you're hyperpolarizing the neurons in the brain and preventing them from firing off action potentials, loss of consciousness. A common drug that we use for this is the drug that Michael Jackson succumbed to because he was self-medicating with it. It's called propofol. What happens is, and it's likely, there's still some uncertainty about this, it causes these chloride channels, and you let chlorine in, you let this chloride ion in, it causes the voltage of the cell to go way low. And if it goes way low, it gets farther away from its ability to fire off action potentials. That's why we call them depressants. They depress the number of action potentials. Now, there are lots of other things about neurons that are super important for healthcare. That includes mood, that includes side effects, that includes muscular function and muscle weakness and other sorts of things. Okay, so the anesthesia is just a beginning. Let's show you some divisions. Okay, let's get with some divisions. Well, first of all, we have location. We have the central nervous system, which includes the brain, the spinal cord, okay? And I often have the brain stem as a secondary structure, right? Yeah, it's part of the brain. Okay, whatever. Okay, it's right in here. It's just this brain and this spinal cord. As soon as you get outside of the brain, like in the case of some of the cranial nerves, or outside of the spinal cord, in the case of the spinal nerves, you are now in the peripheral nervous system. Periphery means outside, out here, out in the appendages, out down the legs, and anywhere outside of the brain, outside of the brainstem, and outside of that spinal cord. Again, a positional division. There is function there, but we've classified them by position. Then you have this third one, and I talked about this in class, the autonomous nervous system, because autonomic, people don't always link up with autonomous. We kind of know what autonomous is now that we have cars driving themselves around, okay? Or autonomous drones and submersibles and all sorts of things. Autonomous means self-regulating, which means your conscious center, you cannot command under normal circumstances. It self-regulates. And in that autonomous or autonomic nervous system, you have the parasympathetic, sounds kind of complicated. It's the rest and digest. All right, and you have the sympathetic, again, which doesn't make all that intrinsic sense when you think, oh, I'm sympathetic to something. No, sympathetic here means fight and flight. Part of why they call it sympathetic, by the way, is because what happens with the sympathetic is you tend to get system-wide responses. Whereas the parasympathetic, you pray you don't get system-wide, because if all your parasympathetics go off, it is super unpleasant. Imagine pooping your pants and peeing and drooling and all sorts of things all at once. Not pleasant. So we tend to have these targeted situations in the parasympathetic, and we tend to have systemic situations in the sympathetic. But I digress a little. Now we're going to get into directional, and now we're starting to get a little bit of function going on, a little bit of function. We have what's called the efferent effector, okay? This is an effector. These are the neurons that transmit signals out into the periphery, all right? The efferent nervous system, also known as the motor nervous system. This is the one that commands skeletal muscle by and large. Okay. So they are going out into the periphery. They are exiting the periphery, if you want an E for you. The sensory, also known as the afferent, is bringing stuff in for processing in the central nervous system. Doesn't have to go all the way up to the brain. Doesn't have to go up to the brain. We can get subliminal signals that are really only mediated in the spinal cord. Okay. Let me give you an example of this. If you do a patellar reflex, you know what happens? You stimulate the knee down here, it comes up the spinal cord, and then goes back down to those quadriceps. Okay. Then you feel it after it happens. Your leg basically kicks itself, and then you feel it. Okay. That's because it was acting at the level, it was integrating at the level of the spinal cord. Central nervous system, by the way, and I mentioned this last night, is an integrating center. You're linking inputs with outputs. All right. The sensories would be the inputs. All right. And the motors would be the outputs. And the central nervous system links those two. Then we have, again, a little bit of, a little more, location. So somatic would be another kind of a word for motor. All right. Very common. There's also some sensory stuff when we talk about somatic. You have somatic senses, like your sense of touch. Okay. That sort of thing. You also have a division of motor that's autonomic. This is mostly involuntary. Okay. What we're talking about here is sometimes the central nervous system will affect some functions, but again, subconsciously, autonomously, without conscious command into the visceral area, digestion, that sort of thing. So let's get into the glia now. Let's talk about the neuroglia, or just glial. So, okay, they're just glial cells. All right, I don't want to get all crazy with these funky words. All right. 4 big ones, and this is the biggest of the big ones. It's called an astrocyte because of its stellar type shape. It has lots of these projections, all right? Lots of these projections. It projects onto the capillaries. Normally, capillaries are super duper leaky, okay? They're normally super duper leaky. Well, the astrocyte will send its foot processes out to the capillary and make those capillaries really, really tight. So tight, what I mean by tight is that so interlinked that material that gets into the central nervous system must pass through a cell. Whether that's the endothelial cell of the capillary or the endothelial cell and the astrocyte, that's the blood-brain barrier. The astrocyte actually sends out signals that create the blood-brain barrier with the blood vessels in the brain. All right, that's one of the functions. All right, astrocytes are the most abundant. They do the most varied set of things. All right, they do support and assist the neurons. They're involved in the synapses. That means they can kind of modulate neuron function. And like I said, they will associate with capillaries and help create the blood-brain barrier. So lots of things. Be able to just recognize these things. Okay, support. They support. Neurons are so delicate. They're floppy. They break easily because of these super long axons. There's a lot of torque on those axons, okay? They are super important in the blood-brain barrier. That's what this play the role in exchanges between capillaries and neurons. It's a blood-brain barrier. They will help guide development and, in some cases, learning. All right. Okay. Control the chemical environment around the neurons. They actually assist the neurons with metabolic processes. These neurons are so specialized and so focused on managing their ions and their neurotransmitters that it's almost like they've forgotten how to process other molecules. And so the astrocytes help with that, which means that they can influence neural functioning, which means that they can help modulate synapses. They can help manage neurotransmitters, and they actually are part of the brain neural processing. We talk about neurotransmitters, We also have these things called neuromodulators. Okay, and we're not going to get into that topic, but that's where the astrocytes really do their thing. Okay, long-term memory, for instance, pretty interesting stuff. Okay, so get to know these, be able to recognize them from a list. Okay, because that's in part how we manage injury in some cases or do therapy. Then we have what are called microglia, or a single would be a microglial cell. Microglial cells are basically modified white blood cells that have migrated out of the bloodstream and differentiated further. And now that what they do is they will help fix injured neurons if the neurons are injured, or if the neuron is going to die, what they will do is they will phagocytose that dying cell. If you remember, I'm trying to remember if we talked about it. If we talked about necrosis versus apoptosis, the problem with necrosis, okay, that's usually the product of trauma or poisoning. The problem with necrosis is that it's inflammatory. It will cause inflammation in a cell that has actively been disrupted and broken up. And the last thing we want in the brain is inflammation. Okay? Nasty, nasty stuff and lots of problems with inflammation. It happens. Sometimes we can't help it, but we want to minimize it. And so what the microglia will do is they will help prevent inflammation by absorbing that neuronal debris through phagocytosis. Okay. Microglia can also help with bacteria or other microorganisms that may have gotten past the blood-brain barrier and into the brain area. Okay, it happens. And they help mitigate infection. Okay. But they're really kind of what we call I like to call them the Roombas of the central nervous system. They will soak up stuff that doesn't belong or could contribute to further injury in the central nervous system. Then we have kind of an interesting population of cells called ependymal cells. Here's some ependymal cells right here. They're kind of funky. They have these cilia, and they help circulate. They actually help create and help circulate central spinal fluid. Central spinal fluid is slightly different than the interstitial fluid, the extracellular fluid we find around tissues. It's a little more acidic. It's got a little more calcium. It's got some other things. And so these ependymal cells, they say they may be ciliated. I think that's the general consensus, that they're not only creating, all right, they're creating cerebrospinal fluid, and they're also helping circulate that cerebrospinal fluid. And then lastly, we have something we've already mentioned, the oligodendrocyte. Oligodendrocytes are really cool. They're kind of look like the Starship Enterprise or something, or with those warp nacelles, okay? What they do is they send out projections to the axons. All right, these are the axons, the central nervous system nerve fibers, and they will begin wrapping these segments around and around and around and around, forming a sheath, a multi-layer cell membrane full of lipid sheath to insulate those axons from each other and to speed up transmission. When we get to saltatory conduction, we'll talk about that. Okay, so here's just another picture showing the different, here are a couple of neurons in here. These have myelinated axons, okay? These are multipolar neurons. All right, well, here's our astrocyte. And again, you can see the astrocyte interfacing with this neuron, influencing it, helping it with metabolism, okay? You can see it attaching and interfacing with the capillary, again, helping seal up that capillary so it's not leaky, creating the blood-brain barrier. Here are ependymal cells. Now, ependymal cells are found in very specialized areas of the brain. There's only a few regions where you find them. That's why we think they help create cerebrospinal fluid. You can also see the cilia here, which means that we believe they help circulate the cerebrospinal fluid. Here are those very, very cool oligodendrocytes, a cool word and a cool cellular structure. Again, they project out and interface with the axons and form these myelin sheets, these little segments of myelin, look like little sausages here. I'll talk a little more about that in a second. And here you can see it's smaller than the others. A microglial cell, a microglia. There's one there, one there. Again, the Roombas. Okay. Let's look at some pictures of actual tissue. And here's a cell, a neuron cell. You can see these kind of projections here. Those are where you would find dendrites. Kind of hard to see in this image. All right. They're very fine hair-like structures that are the inputs. Okay. They bring signals into the cell body for summation at what is known as the axon hillock. Now, right before the axon hillock ends and the axon begins, right here where my dot is, there's a little bit of axon here that is unmyelinated. That's known as the initial segment, the first segment. Then we have a node of Ranvier here, and a node here, and a node here, et cetera, et cetera. And there's some nodes over here as well. Now, axons don't have to be myelinated, and I suspect this axon is actually not myelinated, at least not yet. It should be. I think I just don't see it. But axons don't have to be myelinated. Many are. The myelination speeds up the signaling process tremendously. All right, and call it saltatory conduction. And again, we'll get to that in a little bit. Let's look at one of those really cool oligodendrocytes. So here's the cell body of the oligodendrocyte, big giant nucleus. All right, here's the cell body. And these are all these projections. These are all axons that this or other oligodendrocytes have wrapped up. Now, here in this picture, you see 3 or 4, but here you can see they might do dozens, okay? This dark stain around the outside of these structures is myelin. You can see it's multi-layered. And let's zoom in on that for a second. All right. So here is an axon. All right. And you can see the many layers. There's at least 20 or 30 layers. That's because myelin serves as an insulator. It is preventing unwanted ionic exchange across the plasma membrane of that axon. In fact, demyelinating injuries, things like ALS or multiple sclerosis, for instance, one of the signs that you have something happening is you get tingling. All right, you get inappropriate perception of a signal that doesn't exist. Your brain thinks there's a signal, but it's just happening on its own. Tingling or numbness or muscle weakness because you can't command the muscles anymore. All right, that's the problem. When you lose that myelin sheath, you lose the ability to regulate ion exchange, and that leads to inappropriate depolarization and loss of signal in some cases. Now, in the peripheral nervous system, we have 2 types of cells. One is kind of like the astrocyte. It behaves like the astrocyte, but it looks very different. It wraps itself around the cell body of the neuron. It's supporting that neuron, okay? Maybe a little tricky to see here. Here's a neuron cell body, and these cells that are kind of wrapped around it here are satellite cells. That's why I call them satellite cells, because they're like the moon. They kind of revolve around the earth, if you know what I mean. Okay? Satellite cells are very similar to astrocytes. They behave in the same way. As support, they help with metabolism, may help with signaling in some cases. So they surround the cell body like a satellite surrounds a planet. Okay? Then we have, and here's another picture here. You can tell, maybe, I hope you can see the layers there. This myelin, that's a Schwann cell. This is the Schwann cell nucleus right here. So we've got the cell body there. But what happens is it just wraps itself around and around and around and around. And so you get these multiple layers, increasing the functionality of that insulation that myelin does. Right? It surrounds all the peripheral nerve fibers. Not all of them, because we do have some unmyelinated fibers, like, for instance, we call it the C fiber for pain and stuff. Anyway, they function like oligodendrocytes, but they're also super important for helping damaged axons. If you have a concussion, okay, or if you have a serious appendage injury, oftentimes there's a little bit of neural damage there. Well, your Schwann cells and oligodendrocytes and stuff in the central nervous system will help repair those axons, okay? It's one of the reasons why you want to rest but not fall asleep during when it comes to potential brain injuries. Sleep actually can induce activity. So what you want to try to do is minimize activity you can, all right, and then allow these cells to help regenerate those axons. As we mentioned earlier, there are lots of ways to classify neurons. You can classify them by location, you can classify them by shape, you can classify them by neurotransmitter, etc., etc. All right. Where we're going to do this, we're going to classify them by their structural conformation. Okay. So we have basically three groups. The first one is the most common. It's what we call a multipolar cell. That is to say, you've got a cell body with several dendrites and always one axon. Even if that axon is branched, it is nevertheless a single axon. Okay. Here is also another multipolar with lots of dendrites, but one axon. We call this a pyramidal cell because it's got this kind of pyramid shape. But again, it's a multipolar. Okay, it's a multipolar cell. Again, at least one, it has an axon and at least two dendrites. Okay, so a minimum of three, can have many dozens or even hundreds. So there's a picture of the multipolar cells that I was just showing. Then we have what are called the bipolar. That means you have one axon and exactly one dendrite. Now again, these dendrites can eventually branch out, but they do come in as a single process. Here's another example of it. It's a little harder to see here. Where we see this is in the vision, okay, in your retina. You also see this in the cells that give you the sense of smell. One of the really interesting things about these neurons is they can regenerate. I'd love to learn how to do that with all the other neurons, but be it as it may, we see these in the special senses, particularly in the nose and in the retina of the eye. The third one is pretty much a sensory neuron. It's called unipolar. That's because it has just this one fiber. Okay, it's got these sort of dendrite-like receptors up here, and then it just doesn't even go through the cell body like the other ones. Like, for instance, the dendrites all have to go through the cell body to get to the axon in all these other neurons. This one here, you've got these sort of dendrite-like fibers that condense onto an axon-like structure. That's what we see these. We see these in afferent neurons coming to the central nervous system, okay? That's why we call it unipolar. Sometimes they call it pseudo-unipolar. Why? Because these are receptors. They're just like dendrites, and this is just like an axon. All right, fine. All right. You see this mostly associated with neurons that are conveying impulses to the central nervous system. So we've been talking about these terms up to this point. Let's just formalize this now. And I'll tell you right now, you should know every one of these terms on this picture. Okay, should be able to write those out, know what we're talking about. Can't have a language if you don't know the words, right? Okay, so let's start with the simple one here. Cell body, that's right here, sometimes called a soma. All right. Not the school of modern art. It is the soma. It is the cell body. It does have a signaling role to play, especially in anything but the unipolar neurons. We have the input fibers known as dendrites. Okay. Dendrites essentially increase the surface area of the plasma membrane and allow for more input contacts. Okay. They're basically there to increase the receptive surface area of this neuron. Because you can put a presynaptic terminal right here. See where my dot is? Right there. Doesn't even have to touch a dendrite, and it will send a signal through that cell. Okay. You can put a presynaptic terminal right here, right on that initial segment, right on that right there, and it'll signal. So the dendrites are just increasing the surface area of the input signals. So here's that axon hillock, and right at the beginning here, here's the initial segment. So you see this thing, this big bulge coming out here. It's often very prominent in motor neurons, okay? See it quite easily. And again, it skinnies out here, and that's the first, and then it's a little bit unmyelinated here. All right, that's the initial segment. This is where the decision on whether or not to fire off an action potential down this axon occurs. We call this process summation. Let's say we get a signal that depolarizes the cell. That's called an excitatory signal. But maybe we get another signal over here that's hyperpolarizing, and they cancel out. No action potential. Okay? So that's where this decision is made, whether or not to fire off action potentials. And we got our axon. This one, again, happens to be myelinated. They always show the myelinated axons. Why don't we ever show an unmyelinated one? I don't know. Okay. Maybe they're afraid to be confused. Oh, great. Then we have the terminal branches. Again, this can happen further upstream. You can get some pretty big branches off of this axon. Okay. These ones happen to be right before the terminals, the presynaptic terminals, or the axon terminals, whatever, or the boutons, okay? These guys are where we send neurotransmitter to a downstream cell, whether that's a muscle cell, if this was a motor neuron, or another neuron, or a gland, okay? If I say the word chocolate, okay? If I say the word chocolate and you start to salivate, that's because there's a neuron that went to a gland in your salivary gland and said, hey, let's secrete some saliva, okay? So those are the structures. Get to know them. Get to understand their fundamental functions. We've been going for about half an hour now. I think it's time maybe we take a little break. I'm going to stop here, but before I stop, can you answer these questions? Okay. Do you recall what we were talking about with these questions? If you don't, maybe it's time to take a moment and review the material that we have been talking about. Okay? Write out a sentence, practice it. There's an old joke. If you want to pull a rabbit out of your hat, the first thing you have to do is have the rabbit in the hat. So if you want to be able to talk about myelin, you first have to have thought about myelin and what you're going to say or what you're going to write. Okay? Don't just recite. Think about what you're going to say and say it. Then give that sentence to somebody else and have them, quote unquote, grade it. See how it's going. All right? That's a secret to being a better student, is work with others and never trust yourself. Write it out and then say it if you have to, if you're working by yourself. Say it out loud. Let it go out your mouth and come in your ears. You'd be surprised how different it sounds coming in your ears than it did running around through those synapses in your central nervous system. All right, well, okay, I'm going to stop now. I'll have another video. We'll continue with more neuronal function in a little bit, so I'll see you.